something, these types of things happen. But fortunately, we figured it out, and it's my great pleasure to introduce today Thiru Ramaraj, who's an assistant professor here in computer sciences. And Thiru is originally from the southern part of India, and he did his education and his bachelor's education in India. Then he went on to get a master's at Ball State University in Indiana, and his PhD is from Montana State University, which must have been a very beautiful place to work in 2010. And both degrees are in computer science, so he's coming at genomics from a computer science perspective. For his dissertation, he worked on development and testing of algorithms for proteomics and genomics data. And then after getting his PhD, he went on to become a bioinformatics research scientist at the National Center for Genome Resources in Santa Fe, New Mexico. And he was there between 2010 and 2019. So he was there working for a good time. And then he joined us here at DePaul in 2019, and he started on his fourth year. We're certainly very happy to have him at DePaul, and happy to have him here giving us a seminar on what is a really, as I said to the students in general biology, is really excited area of genomics and pangenomics. So he's going to talk to us about graph-based machine learning approaches for pangenomics.
All right. Thank you, Vincent, for that nice introduction. And thank you all for being here on a Friday, 10 o'clock. So if you go,
dozen people on Zoom too.
Okay, yes. So if you go to CDM SOC, Friday is usually very quiet. Because we don't, it looks like there are some classes going on Fridays here. We don't have any classes on Fridays, so just interesting. Okay, so as Vincent said, my background is computer science, right? So I'm training and education, everything is in computer science. In my PhD, I wanted to focus on bioinformatics. So I decided to work with my advisor on a couple of projects related with proteomics and genomics. So that's where I got introduced to the field of bioinformatics and computational biology. So a couple of problems that I focused on for my thesis was on the proteomics side of things, where at that point, it was really a challenging task of predicting the structure of protein using computational methods so that we can go on to know what the function is, which can then be used for development or whatnot. So along with my advisor, we came up with a couple of algorithms that can take in a protein data band file, a PDB file, for which the structure is unknown, and then try to predict it. And then the other work that I did on the proteomic side of things was do a thorough characterization of amino acid that basically interacts between the epitope and the paratope, which is basically where the antigen binds with the antibody. So we ended up publishing the paper, and I'm still surprised to see that it still gets cited. So it's a basic characterization, but it's fairly looks like fairly useful to the community. Even though the algorithms that I was excited about, it's out there, but it looks like nobody uses it right now because there is a lot of other machine learning and deep learning and AI methods that can just basically do the protein prediction at a much better scale that has developed in the last 10 years. Right. And then I was, this was right around 2008, when the whole next generation sequencing revolution was taking place. And I had an opportunity to do an internship at the National Center for Genome Resources in Santa Fe, New Mexico, where they were just getting started with the first next generation sequencer and generating some data, which had high error rate, which was really, really short, 36 base pairs in length, and then mostly single-end data, right? So it was computationally, it was very challenging to deal with data set in order to take those millions of sequences and make sense out of it, right? So I kind of went backwards. Usually you go from genomics, transcriptomics, proteomics, but I started off with proteomics, and then ended up doing a genome assembly-oriented project with Staph aureus genome data, which was used in my thesis. And then I went on in 2008, so data was starting to come out, and the National Center for Genome Resources, they were actually actively looking for postdocs and bioinformaticians to come on board and work with, be part of their computational team to work on these data sets. So I started my genomics journey there, and I really wanted to be there only for a few years, but I ended up being there for almost 10 years. Santa Fe is a beautiful place, so didn't really want to leave. And then eventually I wanted to get into academia, so I started looking for opportunities, and I'm here at DePaul. So I haven't given a presentation to a biology audience in a very long time. So I'm going to talk about some of the most recent work that I've been working with my collaborators on Pangenomics. So there is a lot of computational methods involved, which I'll try to keep it at a high level, but then hopefully I can emphasize the point on how it's related with the biology and what we can learn from this type of analysis. Right. So the title is graph-based machine learning approaches for Pangenomics. Usually, if Vincent gave a nice introduction, so I'm going to skip this. My lab generally focuses on computation challenges associated with the biological data set. So for the past 10, 15 years, I've primarily been working with sequence data, right? Genomics data primarily, and transcriptomics data as well, but different applications associated with it. And then after coming to DePaul, the last 4 years, I've been starting to look at image data as well. So the first, right, so this data set is basically images of MRI images of lungs that's basically representing some tumor. So the active research area here is trying to come up with computer-aided diagnosis that can take these images and classify if it is benign or malignant, so on and so forth, right? So I'm working with a couple of faculty members at DePaul University and a few from Rush Medical Center as well. And then also, so at Montana State, where I graduated, we have the Center for Biofilm Engineering. It's one of the, one of a kind, at least in the United States. So they do a lot of neat work on biofilms, different types of biofilms, environmental, medical biofilms. So, so they have the traditional way, or the main methodologies they use to study biofilms is just imaging using high resolution microscope, and then basically just analyzing these images to find out about the consortium that is present in these biofilm images. So since I have some expertise at SOC on image analysis, I decided to work with some of my collaborators or faculty members that I knew at the Center for Biofilm Engineering to do some traditional image analysis using computer vision techniques to identify, the structure of the biofilm so that we can know who's there primarily. Right now, they do, the way they look at it is these microscopes that they use come with softwares that they use to analyze the data. Even though they take volumes of images, they just pick one representative image based on which they want to tell their story, and then they publish it. So quantitatively, we can do a lot of neat work with these images. So that's something that I've been working on. But for this talk, I'll focus on genomics data. Right. So just a timeline on looking at the advancement in high throughput sequencing. So the main thing that I want to focus on here is, right, so in 2001, we had the sequence of a human genome, at least the first draft, right? So even now we see versions of this genome coming up with filling in missing information or more genes, whatnot. So it's an ongoing effort. And then, so around 2006, 2007 is when the second generation or the next generation sequencing started, right? So Illumina is a biotechnology company that came up with their first machine that can take a DNA and then sequence it. So their machine, they start off here where you could see really short reads. So the x-axis here represents the read length, and then the yield that you have from these machines. So really low yield and then short sequences, right? And then that's when it all started, and everyone was getting into sequencing to answer some of their biological questions. And that's when I also started working with genomics data. So this is actually nice, kind of shows how things have progressed with the sequencing technology itself, right? So we have reads, we have short reads, but then you can get really high yield with some of these technologies. And then there is some long read technologies here, such as PacBio and MinION, where the yield is not so great, but then you have really long read length, right? So computationally, if you have longer reads, it's easy to work with rather than having millions of short reads, which becomes computationally challenging to do alignment or even put them together into a consensus reference sequence, right? Okay, so I don't know, maybe you have seen the slide. So this shows how much affordable or cheaper sequencing has become. So the slide here basically is showing us up to 2021 what the cost of a raw megabase of DNA sequence is, and then right. So when the next generation sequencing started coming up, we see the cost dropping tremendously, and they always compare it with the Moore's law, because in electronics and computer science, so the Moore's law basically states that every 2 years you're going to get advanced chips and sensors that is going to progressively increase your compute power. But this drop basically does not follow the Moore's law, and it's very dramatic. So that's why they have that line there, to just show how drastic it's become in terms of the price being much cheaper. And this is just generating a human genome sequence, right? So right now, with basically a little less than $1,000, we can get a really high quality genome of an individual using the sequencing technology that we have. And over the years, bioinformatics, computational analysis have also been matured really well, right? So when I was working with this data in 2008, we really needed computer scientists to wrangle with this data, right? So there were some tools, but then we had to edit the tools so that it can perform and give results. But now things have really matured. So if you really want to do a genome assembly, there are softwares that can be run on a small site server, and you can get the genome data, right? And also sequencing was always done in these sequencing centers, right? There were these sequencing centers that did this as a service, but now I know Windsor has one in his lab. So all the in biology departments, PIs have their own instrument, and it's become so straightforward that graduate students run these machines, right, and generate their own data, and then the software everything is in place. You can analyze them, and then look for whatever the biological questions you're interested in. So since 2008 until now, we've come a long way with sequencing. So when I was in the National Center for Genome Resources, almost a decade, so most of the projects involved around next generation sequencing applications. which involved looking at small scale SNPs, single nucleotide polymorphisms, and also looking at large scale structural variations, such as insertions and deletions. Right. So basically, computationally, when we look at it, so there's two main analysis that goes on in here. One, when you have these sequences in hand, you either do an alignment or you do an assembly. So assembly, when I say assembly, it basically means you take these sequences and try to put them together into contiguous sequences, scaffolds, possibly into chromosomes, right? So that's where you would start with. So variant analysis is basically taking the sequences, and then you have this reference, and then you align it to the reference, and then you catalog all the SNPs and see what is different between the samples that you are interested in compared to the reference. The other interesting problem is assembling. So previously, it was just mainly within the realm of variant analysis, but then as sequencing gotten better, reads were longer, the quality of the reads were much better. Everyone for anything non-model organisms, right, for which we don't have any genome sequence. Genome sequence were getting generated, and then when you generate these sequences, there is no reference. We don't have any information, so you have to start everything de novo. So one of the things that you do is a de novo assembly. It is fairly relatively easy when you're dealing with prokaryotes, right? Because the organism is less complex. You can, and it has only one chromosome, maybe a plasmid. So it's the process of assembly was relatively straightforward. But most of the projects that I work with is with plants, right? So plant biologists who are interested in studying different crops, they wanted to sequence their crops. So plant is completely a different beast because of all the biological complexities they have, right? One of the big thing is the ploidiness, right? So, and the genome duplication, repetitive elements, copy number variation. So when you have all those complexities, taking the sequence data that you've generated and trying to assemble them is computationally very challenging. So you have to basically get general strategy was you have a deep coverage of short reads, and then you have some long reads. Even then, you won't have a good consensus sequence for your reference. One other strategy that usually researchers do is go the next step and generate what they call physical maps, which gives you long range information about the sequences in your genome. And then you use that information to kind of bring in scaffolds or contigs together. So our goal is, right, so if we have a organism and if we have 12 chromosomes in it, we want to have an assembly down to 12 chromosomes, right? So the process of genome assembly, I think even after all these years with all the data sets and all the computational tools we have, de novo genome assembly of some plants are still challenging. And then, so once you have the reference, of course, you want to do annotations. So you wanted to go in and see where genes are, structural annotation to identify the location of genes along the chromosomes that you constructed. And then also, once you have identified them, assigning functions to them by doing functional annotation. So I spent a whole lot of time working on that. And different crops, different genomes have different characteristics. So it's not like a one size fits all, right? So even though you are doing genome assembly, things change, right? The strategy, the workflow slightly changes from one genome to the other. And then expression analysis. So this is looking at gene expression, right? So this is basically an alignment-based method. So you have your samples, you have biological replicates, you sequence them, and then you align it to your reference and see what genes are expressed, not expressed, and then you use that information to see why there is a particular disease in one, so on and so forth. And then there are also some non-traditional applications, I would say, where you can look at the epigenome and then also do a ChIP-seq, where if you're interested in looking at protein binding regions to DNA, and then also small RNA analysis. And there are a few others, right? So these are some of the more traditional next generation sequencing applications. And these are, I would say, pretty much streamlined now. There is technologies, there is computational infrastructure that is set up to do all these applications. So this is just wanted to mention, since we are talking about big data, the growth of DNA sequencing. So this is from a paper published by Dr. Schatz, who does a lot of work in this area. He's a computational biologist. So basically, this is showing how much data is going to grow, and right. So based on some of the different characteristics, so doubling every 7 months, which is based on the historical growth, will be here every 12 months, which is based on Illumina, right? Illumina is the company that has these machines that's used to generate sequence data, and then based on Moore's law will be here. So we'll be hitting zettabytes, all right? So we are right now more in the petabyte range. So by 2025, in a couple of years, our data centers will have 1 zettabytes of data available. So that's a lot of data, and computationally, we need tools so that we can learn from the data, right? Okay, so that's a little bit of a background on where things are and what I did a little bit. Now I want to switch focus to my research that I'll be talking about, which focuses on pangenomics, right? So previously we just dealt with genome. Now we have genomes, multiple genomes of the same organisms or related organisms or species. So pangenome, the term pangenome is used to represent a collection of genomic information belonging to multiple organisms or species. And then I will talk about some comparisons between traditional GWAS, right? So genome-wide association studies. So that's an established area where lots of researchers have used GWAS studies to answer some of their biological questions. One of the things to keep in mind is that so GWAS are reference-based. In that I mean, so you have a reference, right? And then the lines or the strains that you're interested in, you sequence them, and then you align those different lines to a reference, and then based on the alignment, you would perform your genome-wide associations, right? So it is reference-based, and I'll talk about why reference-based, why there's a reason to fight reference-based. So in this particular research, I'll talk about what we've been doing to move away from GWAS and bring the pangenome into these type of studies. So we named it Pangenome-Wide Association Studies, PWAS. So this is graph-based. So in GWAS, it is based on alignments. So here we use graph-based methods, and then we are not relying on any reference. So it's a reference-free approach, because we get to use a collection of genomes rather than just one genome, right? And then so we can, once we construct the graph, we can look for frequented regions. So we named it frequented regions, but you can think of these to be conserved regions in a set of genomes, right? So so just to keep it more general, we just did frequented and left out just not used conserved. Because if you look at the pangenome, you can always think about the core, right, and the unique in pangenomes. So the frequented regions kind of represents the core in a way. And then we use these frequented regions into machine learning models and try to get genotype-phenotype relationship for the population. Right. So right quickly, genome-wide association studies correlations between differences in DNA sequences and their phenotypes. We do that based on reference, based on using a reference. So we scan for common variants across a population and then compute the association of these variants with highly observable traits using different statistical methods, right? So you start with your DNA sequences, right? So this could be, 50 or 100 lines or strains that you're interested in, and then you sequence them, and then you have a reference, and then you align it to the reference, and then you identify the SNPs, and then you use the SNPs to basically associate them with phenotypes using different statistical methods, and then you predict the phenotype for those, right? So that's what traditionally it's been done, and it's a very established method. There is a lot of publications that's based on genome-wide association studies. Okay, so coming back to pangenomics, right? So now we know what is genome-wide association studies and how it's done. So I just wanted to talk more about pangenomics. So a single comprehensive catalog of all the sequences and variants in a population, right? So very simple definition of pangenomics. So why we want to think about pangenomics, and different groups are working on pangenomics. So we did see that DNA sequencing is becoming cheaper. It's becoming high quality, right? The costs are going in a downward trajectory, as we saw in those plots. This has paved way to sequence multiple genomes per species, right? So there is several genome consortium that is generating multiple genomes for the same organism. So this is from the International Genome Sample Resource Collection. Basically, it is showing different in different parts of the world the number of human genomes that's been sequenced, right? So we can see there's a lot of genomes, and this is just information from this particular consortium. There are other places or other consortiums that have generated genomes as well. So there is not just one individual, right? We are using one individual as a reference, but if you think about it, there is a lot of complete genomes and annotations of different individuals that are available right now. And then if we look at plants, right, different crops. Sorry if it is really small, but I just wanted to put this table from this paper, basically showing the different species in this column here. And if you look at the number of genomes that are available for that particular species, right? So we have 54, 57. For rice in particular, there is 3,000 genomes that are available, right? So when you have so many genomes available for rice, the question becomes, why do we have to rely on just one reference that everyone is using to do any type of analysis? How can we leverage all the information from these 3,000 genomes, right? So that's where pangenomics comes in, right? So I'm just trying to establish why we want to think about pangenomics and why it's important. So here is an example where. Right. So far in genomics world, we have a reference, right? So for human, we have the latest version of human reference. All model organisms do have a well-established reference that's been generated down to the chromosome level. It has been annotated very well, right? So we are going to be using one of those reference. here to do our analysis. And then, right, so if you have a sample that you sequenced and all these alleles match to what's in reference, then it is great, right? So then your analysis will be fine. You can go on and report your findings. In some cases, right, so when you have another sample that you sequenced and it is, so the red ones are in your sample, but then in the reference, it doesn't really match these alleles, then you are creating a bias, right? So the bias happens because of what we are seeing here, right? And this is, this method is vulnerable to missing or misaligning sequence reads that contain non-reference alleles, like what we see here, right? So we are basically biasing all of our analysis in terms of what we are seeing in our reference, right? This was also brought up in popular press. So NPR had a blog where they mentioned about genetic research has a white bias and it may be hurting everyone's health. That's the main reason, is because right now for all the human genomics that happens, we are just using this one reference, which is from one individual, right? So the article, there's a couple of quotes. It said, if we don't include ethnically diverse populations, we are potentially going to be exacerbating health inequalities. And then by not including diversity, we are missing out on great opportunities to make novel discoveries and to be more inclusive of both population, right? So that's right. So it becomes important, and it is available, right? So previously, 10 years back, we relied on that genome because that was the only resource we have. But now we have more and more genomes. How do we leverage information from that to do everything downstream? So that's where pangenomics comes in. And then the other reason is when we have an individual reference and then we are aligning to it computationally, it is easy, right? So it is just, if it is humans, then we have these 3 billion nucleotides, and then we have our sample that you're interested in, we do alignments, it's fine, it will work fine. But when you think about 10, 20, 30 genomes of humans, then you are doing 3 billion times 20, so that becomes really large, and computationally, how do we handle it, right? So we need tools that can be scaled to a large number of data sets, and then also, right, not doing non-pairwise, because if you take 3 billion and align it to the other 10 genomes, then that's not possible, right? So we need to think about something that is non-pairwise. So these are some of the important motivations that led into looking at pangenomics. And pangenomics is not something really new. Researchers have been thinking about it right from the time when we started getting a lot of genome sequences, right? So if we look at all these lists, so these are all different multiple genome alignment tools that can take in multiple genomes and perform alignments based on some indexing strategy, and then you can see variations at the genome level rather than just doing it from a sequence to a reference. So there is all these tools, but most of them are alignment based. And when at this point, when we start adding a lot more and more data, some of these tools cannot scale to what the data sets that we have. So more recently, there is a consortium. It's the Variation Graph Consortium. So it's a big consortium. There is groups from the United States as well as Europe who are working together on coming up with this variation graph toolkit. So one of the quotes that they had from this paper is, Variation graphs, we can also think of them as graph genomes or genome graphs, graph references, or reference graphs. So they basically encode genetic variation within a population as a graph. And the idea is just using that as a solution to move away from reference, right? So we have information from all the different genomes that we have at our disposal now. So it's a community effort to develop tools to replace those based on linear references, right? So right now we have established pipelines that can deal with alignments and look at genome-wide association studies or catalog SNPs, whatnot. So how do you take that and then bring in where it is not a linear reference, but if the reference is more of a graph, right? How do you do alignments on this graph representation that has information about different genomes, right? And there's been some progress made. There are some tools that we can actually use to do some of this analysis. So this is again from the VG paper itself, where they show different genomes here, where things are, so in these sections things are okay, but then from cases here we see variations from one line to the other, which can be taken into account as well. Just a visual, just kind of show, and then it can always take a different path, and then it can come back to its original path, right? So that's what makes graphs a little bit more effective. So, right, so just some introduction about pangenomics and what has been done and what's been going on. Right now, I just wanted to switch gears and talk about how we are contributing, right? So it's a very complex problem. Lots of groups are working on it. So we are also working on one aspect of it, and we have some tools to do pangenome work. But before that, I want to talk a little bit about some technical terms. So it's a high level, but it's important. So when I go to a few down the slides, if I say k-mer value, how is a graph constructed? So you would have an idea. So it's at a high level, so hopefully it'll be easy to follow. So we want to represent them as graphs, right? So we are taking these sequences and want to represent them as graphs. So one of the graphs that is available is De Bruijn graph, right? So it's named after the mathematician. So it's not something new. De Bruijn graph has been proposed back in the 70s, I believe, but there was no real application that used it. But once DNA sequencing started coming out, De Bruijn graph seems like a organic approach to work with DNA sequences. So one of the main parameters that's involved in constructing these graphs is called the k-mer, right? So you might run into this word k-mer if you're reading a genome assembly paper. So it's basically k-mer represents a substring of length k. So for computational people, when we look at these sequences, we see just stretches of strings, right? So there is, once you bring in the strings, there is a lot of interesting problems that come up, and we can try to solve it. So right from a long substring, you just specify a small substring. So k-mer can be 10, 20, 30, 40, whatnot. So mer is the Greek meaning of part. So simple example, say if we have a sequence or a string, right? So let's say string, and then a former of the string will be ATTC, right here, and then a three-mer will be GCG. So you can basically split a string into several overlapping substrings, right? Depending on the k value. So here is an example. So if you take the same sequence and say if you want to k-merize the string using a value of 3, you will get all these k-mers, right? So it's basically GGC, and the next one will be GCG, CGA, so on and so forth, right? And if you look at, if you use 4, right? So you have these formers based on the strength. So one nice property is if you look at this k-mer and this k-mer, they overlap by k-1, right? So GC and GC overlap, right? So that will be the case for all these different k-mers. So you can then use this information to actually construct the graphs, right? So in this example, I have a 17 base pair sequence string, and then I'm using a k value of 7, and we get all these different k-mers, and then right. So the graph basically becomes graph is basically nodes and edges, right? So there are these nodes, and then these nodes are connected based on some criteria. In this particular case, the nodes are the substrings that we have generated from our sequence, and then they, based on the way they are k-merized, you have this overlap, right? So based on that overlap, you can connect an edge. So in this nice example, if we make all the k-mers substrings as nodes, and then the overlap as an edge, we kind of get something like this. So so this is a de Bruijn graph, right? So now if you have a sequence that is represented in a FASTA format, you can basically use a particular k value, and you can generate all these substrings and construct these graphs, right? And then the graphs will look a lot more complex than something like this. But then you have this graph, then you can process to look for interesting regions, for example. Right. So this is a graph. This is you can also generate a compressed de Bruijn graph. So here, if you just look at this, this is just using the strategy that I said. You can come up with the uncompressed de Bruijn graph for this string. So if you look at it, so CTA can actually be compressed into ACTA. Likewise, ACG can be compressed into TACG. So you can go from an uncompressed version to a compressed version. So this is actually key, because the more number of nodes and edges you have, it's computationally complex. So you want to reduce it. So this is a way of reducing it, right? So right now you can take a sequence, and then you can come up with a compressed de Bruijn graph, and then you can work with these graphs to analyze whatever the analysis is, right? So there are different tools that's been proposed to generate these compressed de Bruijn graph. So we basically have tried some of these methods, and we have the de Bruijn graph. And if you really look at this is actually from a paper. This is a small subsection of a human genome. So they took 3 billion base pairs. They k-merized it, and then they used a program called Graph Aligner to generate the graph. And then this is just a subsection, right? So it's just a complex mess. You can always, there are programs that you can zoom in and look at regions that you are interested in to see exactly what's going on. But if you want to think about these graphs, they are very, very complex. So in our approach, right? So we go from the sequence set, and then we use one of the tools that's already proposed to generate these compressed de Bruijn graphs. And then our method, basically, we named it frequented regions, FRs. So we take these graphs, and then we try to find all possible paths in these graphs, and then come up with frequented regions. So I won't get into the details of it. So basically, we can think of these frequented regions as hotspot regions in this compressed de Bruijn graph that are co-visited by a set of supporting paths from individual sequences of the pangenome. So so this can be considered as a frequented region where you have some nodes and then some subpaths that are connecting those nodes. This could be another frequented region that has a bunch of nodes, and then they they have some paths connecting it. So, and these could be sequences from different genomes. If they have overlaps or if they are similar, right? So they're going to be grouped. Right, so I'm not going to go deep into this. So there are a few parameters that we use. So k-mer is basically the k-mer that gets used at the beginning when we generate the compressed de Bruijn graph, right? So if you use, for example, something like 1,000, then it is too high. You might certainly miss something. If you use something like 100, it is too small, and the graph gets more complicated. So there might be some computational challenges. So selecting k-mer value is tricky. That is not a good way to select it. It depends on the data set. We just have to iterate through a few k-mers to find which one gives the best result. And then the other parameter we use is alpha. So alpha is, we can think of it as a minimum fraction of nodes. So sometimes you have all these nodes, right? The alpha lets you, if you want all the nodes to be exactly similar, then put them together. If you relax them, then you can say, okay, they overlap by 80%, but still we kind of group them together, right? So you can kind of adjust that depending on how you want your nodes to be, how similar or close enough want to be. And then the kappa here basically, say if you have 3 nodes that you want to group belong in a frequented region, and say one has insertion, right? Then the kappa parameter will let you either decide to keep the one there or just leave out because it has an insertion, right? So some parameters to see how we want your frequented regions to be, right? So we can use this this algorithm, and then we can come up with these frequented regions, right? So they are just clustering, right? So when you look at these compressed de Bruijn graphs, they are all these nodes, right? So you start each node to be individual nodes, and then you kind of try to group them hierarchically and come up with these frequented regions. And then the algorithm evaluates these frequented regions based on some metrics, and then it'll keep them. If not, it will not merge them. And then also each one has a support which represents how many nodes, how many edges. So based on that support, we can set a threshold saying that if the support is greater than a specific threshold, then report them as interesting FRs. So if you look at this FRs itself, it will get a whole lot of frequented regions. So if you want to filter them, then just based on the support, we can narrow them to some interesting FRs. Right, so we applied this on a small data set. So we started off with 55 genome assemblies from 48 different E strains. So the total size was around 660 megabase pairs. And just going through the process of generating a compressed de Bruijn graph, identifying the frequented regions, we were able to find out some interesting regions, like identifying some introgression in lines that were not in some translocations. So we were able to biologically identify some interesting regions. So which is good, but we also wanted to apply to a larger data set. So we identified 2 data sets. One is East 100 and one is East 200. So we wanted to try our approach on both of these data sets, but right as of now, our approach works only on East 100. East 200 is too large for our program to handle, so it needs to be scaled up. But we still wanted to give it a try. And these are completely different data sets, though there is no overlap between phenotypes. So we identify these frequented regions, and we basically use them as features to do machine learning in order to predict the phenotype, right, for all the genomes that we had in a population. And one of the machine learning algorithm that we applied was random forest. So keeping to the machine learning approach, so we did a couple of experiments. One, using the frequented regions and feeding into a machine learning method to predict phenotypes. And the other one is generating SNPs and then using the SNPs as features into a machine learning model and trying to predict the phenotype, right? So this is, we are not using any reference, right? It's all completely de novo, but here we use reference. That's how you would generate your SNPs to just see how our machine learning method using the frequented regions from a graph worked with a machine learning model from using SNPs generated using alignments. Right, so these are all some of the machine learning experimental setup, right? So one metric that we looked at is root mean square error. So for every model that we built, we wanted to make sure that this is really, really low, so that we can use this as a metric to compare our approach. So this is how we would do cross-validation, right? So in machine learning, you want to split it into test, validation, and training. And then you would you build your model on the training set, and then you will validate it on this validation data set. And if it is not good, you go back to your training, you tune, and then you apply it to validation. And then if it is all good, you try it on the test set to see how it performs, right? So here is all the methods that we compared. So the first one, I talked about these two. The first one is bringing in the GWAS. So here, right? So here it is machine learning, but machine learning using different features. Here it is completely based on computational methods. We are not doing any machine learning to predict phenotypes. Just looking at the results. So we ran East 100 on using our approach, and basically the values here represents RMSE for different methods. So this is SNPs using ML using SNPs. This is GWAS using SNPs, and then this is machine learning using the frequented regions. And basically, we find our approach to be having the lowest error. And this is even though it's only 5.38 compared to 541, it is significantly different when we looked at the p-values. So in this case, for the East 200, even though our approach gave the lowest error rate, we didn't find it to be significantly different. So we are not sure why that's the case, but just wanted to point that out. And then this is, again, looking at the percent performance improvement using FRML over SNPs ML. And in this case, we see most of them show high performance, get better. But here we see quite a bit that are actually going in the downward direction. Right. So we just got this results, and we are trying to kind of understand a little bit more on why it is not statistically significant and why do we get these different results with different data sets. And this one thing that I wanted to highlight is right, when we generate the compressed de Bruin graph and use it for machine learning, so we use different values of k, and then on this axis here we have kappa, and then on the x-axis we have alpha, right? So those are the three parameters that we use, and the green basically represents good results, the red is bad, and we could see, right, depending on the combination of these parameters, you get different results. So parameter selection is key, and we would only know this by running different experiments and looking at it. And then, so parameter selection is key, and it is very data specific, right? So for example, this box here worked really well for this data set. It might not be the case that using K100, using these values for kappa and the alpha values will work for all the genomes, right? So it's one size doesn't fit all, just because of the nature of the sequence data that we get from different organisms, right? So just wanted to highlight that. And so this, so the East 100 is fine. We can do all these combinations and get these values. But when we think about, okay, what if we have a human 100? right, then running experiments like this will be computationally very expensive. So this is another example where we looked at a couple of different phenotypes and looked at our approaches. So generally, the frequented regions using machine learning with frequented regions tend to perform better in both these cases. Right, and then so far the tool is available and it can be used. One of the things using FRs as features for supervised learning models produced enhanced or equal performance compared to SNPs, because we are seeing some improvement, but it is not much higher than what we would see with SNPs. We are still seeing some improvement. The results were based on FR counts as feature values, as well as the hyperparameter that we selected for machine learning models. And then lastly, the machine learning model hyperparameters can be further fine-tuned for improved prediction over GWAS. Right, so some conclusions, and then like lots of interesting work can be built upon this. So it would be interesting to run models on input data that contains both SNPs and FRs, right? So right now we are keeping it separately. So applying machine learning on using both the SNPs and the FRs and see how the phenotype is getting predicted. And then right now we are just doing it on the nucleotide level, right? So we generate these genome sequences, and then we just get our graphs, and then we generate frequented regions, and then we apply machine learning on those regions. So annotations is not used at all. So bringing in the annotation and see how that changes the prediction power, right, is something that that we are working on right now. And also building the compressed de Bruin graph. Right now there are tools to do it, but when, for example, generating a graph for 5 individuals, it's not possible with the tools that are available right now, at least the ones that we have tried. So that's a bottleneck, right? So building compressed de Bruin graphs for a larger data set. And then identifying k-mer values. So as you go higher in genome size and more number of genomes, we don't have the luxury of running it through 5 or 6 k-mers to identify which one gives us the best results. So some pre-processing to identify k-mer values will be helpful. And then the frequented regions pangenomics algorithm that we have right now works for East 100, but when we tried it on East 200 or East 500, for example, we are not able to get the region. So we are currently working on trying to scale that. And then try different ML strategies. Right now, we just tried one approach. So there are other machine learning models like support vector machine. So trying in a few strategies to see if that helps improve the prediction. And then also visualization, right? So how do we look at it? Right now, it's just a big complex hairball, and there are some tools that can do it, but trying to come up with visualization approaches would be helpful, even though that is really not the core area of our research, but that's something that would be good to have. So my collaborators, so Brendan is my advisor. I still work with him after all these years, and then so these are my colleagues, ex-colleagues at NCGR, and then we have our ML expert, Indika, who's an assistant professor at University of North Florida, and all the work that I presented was funded by a couple of NSF projects. And then, so right now I have a collaboration with Windsor on looking at sequencing of freshwater fish genomes. So that's exciting. When I said yes, I thought I would have some results that I can talk about it, but we are still in the process of collecting data and doing some preliminary analysis. Maybe next time when I come and talk, I'll maybe have some results. I'm generally interested in these areas, right? So DNA sequencing applications, it could be traditional approaches to work with genomics, metatranscriptomics, transcriptomics, more metagenomics, metatranscriptomics, also bringing in machine learning, same thing with image as well. And then one other thing that I'm always interested in working with is trying to integrate omics data. We have genomics and transcriptomics, but not necessarily metabolomics. So maybe if there is data sets, it could be interesting to do some integration studies. I think that is all I had. Thank you.
Thanks for such an interesting talk. We're a little short on time, but just quickly, it's kind of one of these extraordinary things. I'm old enough to remember when it was hard to generate a sequence for one gene. The idea that you're seeing graphs summarizing whole genome data for multiple individuals and species and analyzing that and getting that to details of the nature of life is really extraordinary. So it's just an incredible time to be a student. Do we have any, maybe a quick question? Yes.
What does it mean to bring in annotation?
Oh, so genes. So right now, everything that I presented was just looking at the nucleotide sequences and then generating graphs based on the nucleotide sequences. So if we have a region right now in our graphs that we think is represented in most of the population, we don't know if that's actually a gene or not. So you can think of adding in the annotation information to see if that actually represents a gene, right? So that's what I meant, bringing in the annotation to see if there are any genes involved. So so the VFRs, so you get this nice output where you have the genome ID, what is the chromosome, the start and end of the chromosome, and the sequence itself. So we have all that information. So now we can go back, and all these genomes are annotated. So we have GFF files for all these genomes. So now can we overlay that on these bed files to see if they are associated with genes? And then the other approach is just looking at just generating graphs of these genes, right? Rather than the whole genome itself, and then doing similar analysis and see how that can be used in predicting, going from genotype to phenotype prediction. That's what I meant.
Yeah, and in biology, we're often used to, when you're doing an analysis, just you press a button. So next time you press a button to do an analysis, like sequence alignment, you can think of Thiru's work and say, thanks for doing the hard work. That lets me just press the button and get the result. Thank you for a wonderful talk.
Yeah, no problem. Thank you so much.
Thank you, everyone. There's some treats in the back if you have a few minutes, and thanks for everyone for coming.